vascular system. By the way, just so everybody knows, for Monday, we might, or Tuesday, we might get into it a little bit today, but I doubt it. Today we're going to talk about a little bit of the anatomy of blood vessels. And then we're going to get into, from that point, dealing with just a tiny little bit on lymphatic system. I am not going to ask you guys to read the chapter on lymphatic system because it really goes into a whole lot more depth than what a course like this is really going to allow for. So your reading assignment would be the chapter on the respiratory system. And to that end, you're going to do what you've done now multiple times. You're used to this. One of the things you're going to do is trace the path that air takes from the point that it enters the body until gas exchange occurs in the lungs. That's gas exchange with the blood. Naming everything that the air passes through in order, and basically what's happening to the air as it passes through there. The chapter on respiratory system is not terribly long. Some of you have a little bit more time on weekends to study than on other times. So if you're looking to get a little bit ahead, come Thursday of next week, we're going to do digestive system. And in honor of the new McDonald's that they put over across the road here, so that we can have pseudo food close to campus, you will take and trace the path that a Big Mac takes from the point that it enters the body to the point it passes virtually unchanged because they're indigestible when it leaves the body. And basically what's happening in all of those structures. Okay? That way you won't be getting bored. Because I know you guys don't like things. All right. If you all would, get out your book for a moment. I'm going to refer you to an illustration here. Dealing with blood vessels. Oops, all the way to the digestive system. That's all right. Dealing with blood vessels. There is an illustration I'd like to refer you to. I do not have any good models for this in the lab, which is unfortunately true. But I want you to be aware of this because, again, especially those of you who are moving on into, I don't know, physiology and things like that, you're going to need this. Have a look at page 572. On page 572, you see a diagrammatic representation that shows you different kinds of blood vessels and the components that make up their walls. All right, as you look at these, look over on the right, your right-hand side, where you deal with arteries. Gang, there's really two major kinds of arteries you deal with in the human body, elastic and muscular. To understand the difference between these, you need to understand the basic layers. Where they have the elastic artery layered out or labeled out there, you see the outermost covering is what's referred to as the adventitia. In some older textbooks, they call this the tunica adventitia. Tunic means jacket covering. So adventitia layer is fine. Adventitia layer is going to be made up, for the most part, of dense irregular connective tissue. The next layer deeper to that is what they're labeling the media layer. And the media layer, in virtually all blood vessels that have this, is going to be made up of single unit smooth muscle. It's going to be made up of single unit smooth muscle. And then you have what they refer to as the intima layer. And here, the intima layer is going to be made up of the endothelium, which is going to be simple squamous epithelium, And you see additional connective tissue in the internal elastic layer. This would be a tissue they call elastic connective tissue. All right, if you compare that elastic artery to the muscular artery, great big huge monstrous differences you see. First off, you don't see that elastic component in the intima. The intima layer is much thinner in a muscular artery. Secondly, the medial layer in the muscular artery is much thicker compared to the diameter of the artery. Most of the arteries in the human body are muscular arteries. A very thick medial layer. The only elastic arteries, the main ones that we deal with in here, would be things like, I don't know, the aorta is an elastic artery. The pulmonary trunk is an elastic artery. They're usually going to be relatively large arteries fairly close to the heart. The rest of the peripheral arteries are mainly going to be muscular arteries. Reason for this, we'll see here in just a moment. Go down to the level of the arteriole. When you look at an arteriole and compare it to either of the other kinds of arteries, arterioles are very small. They're very nearly microscopic in size. And take note, you have the medial layer, no adventitia, and you have the intima, and that's it. The smooth muscle component is directly exposed to the extracellular fluid. You don't have that connective tissue covering. Capillaries, when you look at a typical capillary in the human body, all it has is the intima layer. And really, a typical capillary is only going to have the endothelial part of the intima. So in other words, it's going to be made up pretty much of simple squamous epithelium. Capillaries, by their very definition, are microscopic in size. Very, very tiny.
So the endothelium, that is the...
Endothelium, in this case, is simple squamous epithelium.
Okay.
All right, going back up the other side, your left side, we have venules. And if you look at a venule, you see an adventitia and that endothelium of the intima, and that's it, no medial layer. If we move up into medium, actually small and medium-sized veins, you have an adventitia, a media, and an intima. But if you note, the media is relatively thin. Nothing like you see in the muscular arteries. Large veins, again, you have all three layers, but you have a very thin media layer. The adventitia layer is actually the thickest layer of it. Okay, these aspects of structure you have to have a little bit of a handle on in order to understand how circulation works in the body. Figuring out how blood moves through the arteries and veins in the body is not complex anatomy or physiology. It is plumbing. Plumbing 101. What I'm about to tell you is something that anybody that installs sprinkler systems has to know. Or anybody that puts plumbing in a house has to understand. It is not high-end physiology or anything of that sort. Flow of blood through the blood vessels is going to follow a pressure gradient. It's always going to go from an area of higher pressure to an area of lower pressure. So in order to get blood to continuously move through systemic circulation, we're going to start at the heart, we're going to end at the heart, right? You're going to have your highest pressure as blood leaves the heart, and the very lowest pressures as it comes back into the heart. You have to have that pressure differential, or the blood's not going to move. All right. The arterial side of circulation. In physiology textbooks, they refer to it as a pressure reservoir. where we kind of store pressure in the system. The venous side of the system is going to be more of what they would call a volume reservoir, where we store excess volume of blood at need. Okay, how can we have a pressure reservoir? You all know that contraction of a chamber of the heart was called what? We gave that a name. What? Systole, good. Contraction of a chamber of the heart is systole. How many people here have ever had somebody take their blood pressure? Oh, good. I would expect everybody's hands to go up, because at one point or another, hopefully you've had that done. That's part of any normal physical examination. You know, when they take blood pressure, there's two numbers that they usually give you. Yeah? They give you a systolic pressure and a diastolic pressure. First off, they are measuring pressure in systemic arteries. That's specific. We don't care about pressure in the veins because they're low pressure vessels by definition. We have no way to really measure pressure accurately in those, at least not without getting invasive. So they measure pressure in the arteries on the arterial side. Why in systemic circulation? Well, King, you have to have access to an appropriate artery. You know, the cuff that they put around your arm, they call that a sphygmomanometer. They're compressing an artery to the point blood won't move through it. If they wanted to do that in like pulmonary circulation, they'd have to use a much bigger cuff and put it all the way around your chest and crack your ribs as they compress the chest to the point that the pulmonary arteries were closed off. At which point they wouldn't get any pressure because you would be dead. So that's not going to work. And likewise, trying to put one of those around the heart and compress the heart to the point blood won't flow through coronary circulation, you run into similar problems. You're going to crush the heart doing it. The only way we conveniently do it is with systemics. All right, so we need systemic circulation. And that systolic pressure then, since we're dealing with systemic circulation, that's the high pressure. That would represent pressure that results from contraction of what specific chamber of the heart? What? The left ventricle. Yeah. Only the left ventricle. We're measuring maximum pressure generated by contraction of the left ventricle, which is why they call it systolic pressure. Systole specifically of the left ventricle. The diastolic pressure is a lower number. If it's not a lower number, there's a real significant problem somewhere, and you should probably be in a hospital. The diastolic pressure is a lower number, and that is the bottom pressure, the lowest pressure you get to in the vessels at the end of diastole of the left ventricle. To make blood flow work, we need these elastic arteries, because the ventricle isn't always in systole, yes? We need to have something continually applying pressure to move blood on through the rest of circulation. So between ventricular systoles, these elastic arteries, you get this big surge of blood volume going into the elastic arteries, they stretch. That elastic layer in the tunica intima allows them to stretch to a considerable degree, and even more importantly, when the ventricle goes into diastole, because you're dealing with elastic connective tissue, it's going to rebound back to its original shape. And as it does, it continues applying blood to the pressure, or to the applying pressure to the blood in these elastic arteries, and they've only got, since the semilunar valve is closed, there's only one direction of blood can move, and that is on further into the arterial aspect of the system. This is how we maintain enough pressure to keep the blood moving between ventricular systoles. It's not like the blood is going to completely stop between systoles. It doesn't. You've got continuous pressure being applied by the elastic component of the big elastic arteries. All right, so that maintains pressure in the system. Now, we got another problem here, and that problem is this. When you guys did blood vessels on the heart, you might have observed this. You're doing systemic blood vessels in lab today, those of you who haven't had lab yet. And you'll see it, those of you that were in lab this morning have already seen this. But all of you have looked at the coronary circulation. Did you notice that the further we get from the origin of the coronary arteries, or vessels in arteries in systemic circulation, the further away we get, the smaller in diameter they become. Okay, there's reason for this. There is friction that occurs between the blood and the wall of the blood vessel. And it's something that people have trouble wrapping their heads around a lot of times. They think about pressure, and they think, well, hey, this whole pressure thing doesn't work for me. I'm sorry, friction. They think about friction. They think this whole friction thing doesn't work for me. How can liquid have friction? It does, very much so. Anybody here ever go like on a float trip? You know, like on a river? Okay. The part of the river that's going to be moving the fastest is going to be the part typically that's deepest. If you get out in the middle of the deeper part of the river, the canoe will drift along faster than it will if you're up near the bank. Why? Well, you've got a lot more volume there with less of it in contact with the bottom or the edge of the bank. Literally, if you drop like a leaf in the water in a river right up against the bank, a lot of times you'll notice a leaf dropped 4 or 5 feet out, passes it up, moves faster. This is because of friction with the bank. Sometimes the leaf against the bank actually looks like it's moving backwards because of some eddy currents that develop as a result of this. Okay, movement of blood through your blood vessels can be very much like this. The larger the diameter of the vessel, it gets into that whole surface area to volume ratio thing again. We've got less blood actually in contact with the walls. So in the center, the blood moves faster than it does up against the outside walls. But the problem is you still have friction going on, and the longer the vessel is, the more pressure and velocity you're going to lose because of that friction. Those of you live in a house, have to go out and water the yard. If you have a relatively short garden hose and you hook it up to the spigot, turn the water on, water comes out of it with pretty substantial force. It's all the way on. If you take and hook a real long garden hose onto it, the force that the water comes out with is less. If you hook several garden hoses together, the amount of pressure you get coming out of the garden hose at the end is even less. You're dealing with loss of energy due to the friction in the walls of the garden hose. Blood vessels work the same way. In plumbing, the only way to maintain pressure over a long pipe run is to decrease the diameter of the pipe as you go further along. That keeps pressure up. You put a nozzle on the end of your hose, and you get the water to come out with a whole lot more force. Why? You're decreasing the diameter of the opening the water's passing through. So a lot more pressure builds up. In the arteries then, the further you get away from the heart in systemic circulation, the smaller the diameter of the artery that you're going to be dealing with. Be a gradual decrease. But we've got to also think about volume. We've got to carry the same volume. So in order to do that, the arteries are going to branch. And you're going to have more and more arteries that you're going to be working with so that you can still carry the same volume. Okay, this is where we get into the muscular arteries. If we want to vary the amount of friction, the amount of resistance, the thick muscular walls of these muscular arteries, and gang, I have seen muscular arteries in cadavers. The elastic arteries, you take all the blood out of them, they just collapse, because they don't have much of a media layer. Muscular arteries in a cadaver, in many cases, will be pretty firm, even with all the blood removed, because that much thicker media layer. Thicker media layer means you can vasoconstrict more, so you can adjust pressure a bit more. All right, so they're not going to have the elasticity of the elastic arteries, but there's going to be a lot more of them because there's a lot more branches to them, and they're going to be running, again, at relatively high pressure. So, basically, it's going to work kind of like this. From the heart, start out with big elastic arteries, and those arteries are going to go through branchings that branch into progressively smaller and smaller diameter muscular arteries. And all the time, these smaller branchings are increasing resistance so that we maintain pressure. Do we lose some pressure? Absolutely we do. Through this system, there is going to be a pressure drop the further we get from the heart. There has to be, to keep the blood moving. Eventually, we get these arteries branching into almost microscopic branches. That these are what we call the arterioles. Arterioles, from that diagram in your text, you have a media layer that's not terribly thick, and an intima, and really pretty much nothing else. This is where the largest single drop in pressure is going to occur in systemic circulation. The arterioles are where the biggest pressure drop is going to happen. Now, our arteriole, then, is going to branch into multiple small branches, very, very tiny branches, and these would be the capillaries. Gang, capillaries are so small in diameter that in most cases, erythrocytes, red blood cells, have to pass through them single file. You can't put two erythrocytes side by side in most capillaries. So these are incredibly small blood vessels. When they first started looking at human circulation seriously in Europe, one of the things that they determined was they saw the blood vessels got smaller and smaller and smaller as you got further away from the heart, and then they just seemed to disappear. You go back into the 1400s, they thought that blood just vanished at that point. It went away. Somehow it magically just disappeared and you were constantly producing new blood. A man by the name of William Harvey, 200 years before the microscope was invented, proposed that there were tiny little blood vessels that connected veins and arteries, things that he called capillaries. Never having seen one, he died never having seen one. These were theoretical structures that connected veins and arteries, too small for the eye to see, and that blood recirculated through the body. You had continuous circulation going on. Okay, Harvey was pretty much a laughing stock for a while because this was just totally ridiculous. Everybody knew the blood just disappeared. Seriously, that was the way the scientific community looked at it at the time. Harvey was later determined to be correct, absolutely correct in his assumptions, that we do have these tiny little capillaries that interconnect blood vessels and have continuous circulation of blood. These capillaries are just incredibly small in diameter, and this is where, since we only have the intima layer and just the endothelium, simple squamous epithelium. You guys looked at simple squamous epithelium from an edge view. It is rather incredibly thin. When we talked about that, I made it a point to tell you that this was where you didn't get a lot of wear and tear, and where you wanted to do rapid exchange of materials from one side to the other. Okay, in the capillaries is where we have exchange occurring between the blood and the surrounding tissues. Gases, cellular waste, nutrients, all of that stuff. The simple squamous epithelium of the capillary walls allows for fairly rapid exchange. And you have some aspects of plasma that are going to leak out of the capillaries here. Now, not all of the plasma. The larger proteins and whatnot are retained. But water and smaller molecules that are suspended in the plasma will be allowed to pass from the plasma into the interstitium. Gang, we've talked about extracellular fluid or interstitial fluid throughout the semester. This is where it comes from. Interstitial fluid or extracellular fluid is filtered blood plasma. Stands for filtering things based on size and using pressure. The process is called filtration. It's a passive process. So we're going to have some, not all, but some aspects of plasma leaking out of these capillaries into the interstitial spaces between the cells. And again, we call that filtration. As we move through the capillary bed, we still get a drop in pressure occurring. As we move through the capillary bed, our capillaries are going to start essentially growing toward one another, and they're going to eventually fuse to form very tiny vessels that we call venules. On this side of the capillary bed, we're going to have some of this interstitial fluid move back, because the pressure in the interstitial fluid is lower than the pressure in the blood at this point, or I'm sorry, higher than the pressure in the blood at this point. Some of this is going to move back, and this is what we call absorption. In capillary beds, you're going to have both filtration and absorption occurring. Interesting thing is, filtration always occurs at a higher rate, faster rate than absorption. You can consider that virtually a law of the way capillaries work. Filtration in a capillary bed will always occur at a greater rate than absorption will occur. All right. Our venules are then going to come together to form larger, and as they're joined by other vessels, even larger veins that are going to carry blood back toward the heart. In the venous aspect of circulation, since blood is moving back toward the heart, you're going to have smaller vessels coming together to form progressively larger and larger vessels. And again, the reason for this makes perfect sense, because pressure has to progressively get lower the closer we get to the heart. To get that pressure drop to occur, as we have all these smaller veins coming together to form larger and larger veins, hey gang, we've got more volume we're dealing with. The only way we can get larger volume moving through these and continue the pressure drop is to have the vessels get larger in diameter, until eventually we get into the vena cava. Yeah? Something else we see in veins, and this is completely different from what we'd see in arteries, and that is, in our veins, we're going to have periodically internal valves. Okay, if you think about the semilunar valves that you guys looked at associated with the heart, the internal valves in veins are very much like the semilunar valves that you guys looked at in the heart in structure. They're made up of connective tissue. They respond to pressure changes, and they only open one way. The deal with these internal valves in the veins is as blood flows toward the heart, it forces the valve open. Then if blood tries to flow backwards as pressure drops, that forces the valve closed, and it keeps the valve from, or keeps the blood from flowing backwards in the veins. So it's like what they would call in plumbing a check valve. Ensures one-way flow of blood. You do not see these in the arteries because there's only one direction blood can flow because of the high pressure. In these low pressure vessels like the veins, we need these kinds of things. You know, there's not much of a tunica media in any of the veins. Veins then tend to stretch a bit more easily. When you are inactive, like now, I mean, none of you guys are really physically exerting yourselves. Or I hope this doesn't count as major physical exertion for anybody. Because you're just sitting there ready. Yeah? In low activity circumstances, a lot of the peripheral veins will dilate. Blood doesn't stop moving in these. They call it pooling of the blood, but it's not really stopping the blood from moving. But the larger, as they dilate to larger diameters, it makes the blood move more slowly through them. Which means blood's getting to the heart at a lower rate. Don't really see this happen in arteries, but we do see it happen in veins.
So when they dilate, it slows down blood flow?
Yeah. Which means the heart's receiving less blood than it does when you're being active. Well, you know, even when they're dilated, the valves still work, at least under normal circumstances. But once in a while, you run into a problem where a valve fails. We see this problem where people stand in one place a lot, especially on a hard floor. And you'll get valves fail in superficial veins and allow some blood to back up, causing other parts of the veins to dilate even more than they would ordinarily. And you know, when you look at a vein through the skin, it looks kind of dark. And you start seeing some of these veins become very prominent. It's what they call varicose veins and spider veins and things of that sort. It's a result of one or two valves failing and the blood literally being able to shift backward a little bit. Okay, last week when we talked about Starling's law of the heart will pump out as much blood as it receives.
Mm-hmm.
We talked about you stretching in the morning, yeah? Okay, now this should make more sense. When you're asleep at night, a lot of the peripheral veins are in a pretty dilated state. When you start contracting skeletal muscles, those veins get compressed, and you get this big surge of volume of blood to the heart, causing the cardiac output to increase. Pumps out as much blood as it receives. More blood goes in, more blood gets pumped out. That's what that's all about. It's not the blood stop flowing to the heart. That doesn't happen. It's that you just slow the blood flow down. So the heart's not pumping as much out. It only pumps out as much as it receives. Yeah? Oh, well, situations. One of the things you'll notice are even out of clinical situations, people are into athletics. If somebody's very active, especially doing aerobic kind of exercises, endurance type of exercises. You know, you get somebody who's a good athlete, and their resting heart rate might be down in the 50s. Yeah? Does it mean they're not pumping as much blood? No. They're pumping at least as much blood as your heart's pumping at the same activity level. The difference is they're getting more blood to the heart. Why? Muscle tone. They've got better tone in their muscles, therefore more muscles are remaining in a partially contracted state, keeping those veins compressed. You get a larger volume to the heart. That's why their heart rate's low at rest. They're working more efficiently. If your idea of strenuous exercise is watching Wheel of Fortune and participating where nobody can hear you on television, then probably your resting heart rate's a bit higher. You're not receiving as much blood in the heart. To keep enough blood moving, circulated properly, the heart has to beat faster. To do the same thing, you're making the heart work harder to do the same job. Yeah?
What you said about muscle tone.
Well, the skeletal muscle tone means that the skeletal muscles are partly contracted more. Okay, that means you're compressing veins more, which means a larger volume of blood is getting to the heart at any given time. So when you're at rest, the heart's receiving more blood, so per beat it's pumping out more. It doesn't have to beat as often to do the same job. It doesn't have to work as hard. That's what that's all about. But now that we've got a little bit of background in how the vessels work, that part of it starts making more sense. Now, you guys are scientists now. You had the science fairy whack you on the head with a meter stick as you came in, and you magically became scientists when you entered into this class. That being the case, when something sounds just totally wrong, you should be, hey, wait a minute. And I said something earlier that should have sounded totally wrong to you. What?
I don't know if this is it, but something did sound wrong. That may just not be me understanding. More vein pressure or more blood pumped to the heart seem like the resting heart rate would be higher.
No. When we talked about the way the sarcomeres in the cardiac muscle work, there's more overlap between actin and myosin. So you can stretch them further and still get contraction to occur. But when they're stretched further, they generate more force.
Okay.
So when you get an increase in blood volume to the heart, that stretches the chambers more. They pump more forcibly. Each time a systole occurs, you get a larger volume of blood being forced out.
Okay.
If you're dealing with restricted flow of blood coming to the heart, or not as much blood per unit time, then to keep your blood pressure up and everything, the heart has to beat faster, more beats per minute to keep pressure up where it needs to be. But what did I say earlier that sounded off? If you say everything, you fail. You fail.
Well, I had a question. If the filtration always happens faster than the absorption, how does that work?
And that's the point. That's exactly what I was getting at. If filtration always occurs faster than absorption, you would expect what to happen? A buildup of
Interstitial fluid.
interstitial fluid, fluid between the cells. more of it's coming out of the blood than is going back into it. Has to go somewhere, yes? Oh, well, this is a good kind of segue. We are almost out of time, but just to give you an idea. You guys have all probably heard about lymphatics before, the lymphatic system. And when most people hear about the lymphatic system, they think about what? Immunity, yeah. Think about different aspects of immunity, because yes, the lymphatic system plays a big role in that. What they don't think about is something that's actually equally important. And that is the lymphatic system picks up that excess interstitial fluid and drains it off. You have in the tissues lymph capillaries. These are just small, dead-end lymphatic vessels that have pores in them. As interstitial fluid builds up, it passively flows into these lymphatic capillaries, and lymph capillaries join together to form larger and larger vessels that we call lymphatics. Lymphatics have no media layer at all. There is no muscle in any of them. They do have an adventitia and an intima layer, and like veins, they have the same kind of internal valves that only allow the lymph to flow one direction. Okay. We're going to take and pick up that excess interstitial fluid and eventually, by way of the lymphatics, dump it back into the blood. How important is this? Next time we meet, I will bring a photograph in. An illustration in the zoology text. In the tropics, in certain parts of the tropics, there is a parasite. It's kind of roundworm they call the filaria worm. It's transmitted by the bite of a particular mosquito. These things get into your blood and migrate into the lymphatics. That's where they really like to be. And if you get enough of them in a given area, they can occlude or block the lymphatics. The disorder is called filariasis, and I don't know that they've ever found a way to treat it. Filariasis goes under a common name that many of you may have heard before. It's also called elephantiasis. Because as the lymphatics block, interstitial fluid builds up and you get edema. You get edema on a level none of you have ever remotely experienced. You get so much fluid buildup, like the photograph in the zoology text shows a man that had it happen in his leg. And one leg was like 3 times the diameter of the other, with big folds in the skin. That's why they call it elephantiasis, where it's all fluid buildup, because the lymphatics can't drain the excess interstitial fluid properly. If the system doesn't work, that's the kind of thing that happens. It is incredibly severe. So, next time, we'll start out talking a little bit about lymphatics, and then we'll jump into respiratory system.